Hello everybody. In the earlier class, we studied grain storage. Now, in this class, let us study disinfestation of grains. There are different ways by which grains can be disinfested, like physical methods are there, chemical methods as well as biological methods. The physical methods generally are costlier process, biological methods are not very useful for bulk storage of grains, they can be done for small quantity grains etc. So, the most commonly used methods of disinfestation of grains include chemical methods, that is here the contact of the grain, infested grain with the some chemicals, insecticides, or it may be a gas fumigation. But these chemical methods also they have certain disadvantages, like they are effective on fully developed insects. They have very little or almost no effect on the eggs or larvae of insects. Insects become resistant to the use of chemicals, particularly that insecticide etc. upon repeated use. So, toward the later stage or later on, it may be after long time, sometime the chemical effectiveness of the insecticide to that particular insect may get reduced. Also, some residues of the product, though not highly toxic, may linger on in foods for some time. So, these are some of the drawbacks of chemical insecticides or fumigants, and therefore, there is a continuous hunt for new fumigants which are environmental friendly, which are highly efficient on all the stages of insects, which have no cross resistance to existing fumigants, which are effective suppression of progeny. They cause effective suppression of all the forms of insects, whether it is larva, egg, pupae and so on, and the process should have minimal effect on the grain induced quality, and ultimately it should be cost effective. So, in this regard, ozone becomes a very potential fumigant for use in grain for the killing of insects or for disinfestation of grains. Ozone, I hope you all know, is a trioxygen. It is an inorganic molecule with the chemical structure O3. Ozone is created in the atmosphere when the sun's rays split oxygen molecule into single atom, and this single atom oxygen molecule or single oxygen, it combines with the oxygen to form a 3 oxygen molecule which is called ozone. As you can see here in this figure, that is single oxygen, it combines with 2 oxygens, and ultimately ozone having 3 oxygen molecules are formed. Having 3 oxygens are formed. So, why ozone or why it is being promoted? Because it is one of the most powerful oxidizing agent. It instantly destroyed microbes and insects. It is environment friendly. It has higher oxidation reduction potential than that of general chemical oxidants like chlorine etc. It does not affect product taste, and it is not harmful to anyone, and it does not give harmful byproducts. So, In this slide, I have just tried to give you the milestones in ozone treatment of food products. In the year 1893, ozone was first time used as a disinfectant in drinking water. In 1909, it was used as a food preservative for the cold storage of meat. In the year 1939, it was discovered that ozone prevents the growth of yeast and mold during the storage of fruits. In June 2001, Food and Drug Administration of the United States approved the use of ozone as an antimicrobial agent for the treatment, storage and processing of foods in gas and aqueous phases. Same year, in the month of December, the US Department of Agriculture, Food Safety and Inspection Service, which is commonly called as USDA FSIS, approved the use of ozone in contact with meats and poultry. So, ozone is now seen as a very effective alternative to chemical and other fumigants in the grain and other food materials, because it is considered to be a non-thermal method. It results into the maximum retention of nutrition, that is disinfestation by the ozone does not have any significant effect on the nutritional components present into the food. It has worldwide acceptance, provides good safety attributes to the treated materials, maintains quality and health attributes, and of course, more importantly, it is considered to be an environment friendly technology that does not cause any problems of pollution, etc., to the environment. So, before I come to the application aspects of ozone in grain storage or other food processing, let us consider that properties of the ozone or structure of the ozone. As you can see here in this figure, that is the ozone is a triatomic oxygen formed by addition of a free radical of oxygen to a molecular oxygen. It is partially soluble in water and its solubility varies with temperature. That is, the solubility in water varies with the temperature. The half life of ozone is about 20 to 30 minutes in distilled water at 20 degrees Celsius. It is a potent antimicrobial agent against bacteria, fungi, viruses, protozoa, and also it is effective against the bacterial and fungal spores. So, all sorts of microorganisms including their spores can be inactivated or can be destroyed by ozone. In this table, detailed characteristics or properties of the ozone is given, like it is a bluish gas or dark blue fluid. Its molecular weight is around 47.99 gram per mole, boiling point about 111.9 plus minus 0.3 degree Celsius. It has a melting point of 192.5 plus minus 0.4 degree Celsius. Its critical temperature is 12.1 degree Celsius, critical pressure is 54.6 atmosphere, and then density, heat of formation, oxidation strength, etc. Its solubility is water at 20 degree Celsius, its 3 ppm is soluble, and it has a specific gravity of 1.658. So, this now how the oxygen can be generated, and since in its properties you decide that it has a very less half life, that is about 20 to 30 minutes. So, and also it cannot be stored, ozone cannot be stored or conveniently purchased by gram, pound, gallons, or tons, etc., like other gases. You can store oxygen in a cylinder, carbon dioxide etc. in the cylinder, but ozone cannot be done like that. So, it must be produced on site as needed, where needed and when needed. So, there are different methods for the ozone generation or production of ozone, and those include corona discharge method, electrolytic method, photochemical method and radiochemical method. And among all these, the corona discharge is the most acceptable and cost effective method which is used commercially for generation of ozone for various food and other applications. The photochemical method of in ozone formation you can see here in this picture. In fact, it is the natural production of ozone either by lightning or by UV radiations. That is the solar light, that is the UV rays etc. of the solar radiations. They split oxygen molecule, you can see here in this figure, that is O2 into O plus O, and these O acts or combines with another O2 and gives the O3 molecule, and then a new O is formed. This new O again this gives. Similarly, this by lightning etc. also, that is they may cause splitting, they may split the O2 into O plus O, and this O individual O they can combine with O2 and give O3. So, this is how this photochemical method results into the formation of ozone naturally. In the corona discharge method, in fact, as you can see here in this figure, there is the dust free, oil free or an oxygen containing gas mixture or even pure oxygen is passed through the space of a high energy electrical field between two electrodes, and these two electrodes are separated by a dielectric material and which is usually a glass. So, the one electrode which is high tension electrode, the other electrode is a low tension electrode, and there is a tube through which the oxygen is passed or discharged, and the dielectric this high tension electrode and low tension electrode, the dielectric material glass is provided here, you can see here. So, this O2 in this discharge tube is converted into O3 and the heat released is dissipated from the system. So, this is in fact one electrode is a grounded medium and the other is a dielectric one, that you can see here in this picture. So, in this way the ozone is generated from the O2 gas or pure gas mixtures. The ozone formation reaction during electrolysis of water can be seen here, like water, when electricity is passed into this, then it gives O2 and hydrogen ions. This if 3 molecules of water, they give O3 plus 6 hydrogen ions. H2O plus O2 gives O3 plus 2 hydrogen ions. So, in this way, that is by electrolysis or electrolytic splitting of water, ozone is generated. The radiochemical method has high energy irradiation of oxygen which produces ozone. This is not yet used commercially for portable or wastewater treatment. In the future, there may be conceivably good use of a excess fissionable material, but currently there is much more fear of irradiation than that of bad water. There is some fissionable materials are required for production of ozone in this process. So, the excess fissionable material is thus considered to be just a hazardous waste instead of a potentially valuable resource. Maybe in the days to come, this can become popular when the radiations hazards etc. are taken care of, or it is made sure that by radiochemical methods, that is ozone will be generated, and the generated ozone is safe from radioactivity point of view. So, the components of an ozone generation unit, as you can see here in this figure, it contains an oxygen tank, ozone monitor or analyzer, ozone generator chamber, and then a fluorometer. So, in fact, this is a setup where ozone generated from this system, it is allowed to pass through in a assembly or in a column which contains orange juice or some other samples. So, that this type of setup can be made in the laboratory for the ozone treatment of the food material. We have also in our laboratory a similar setup, that is the ozone generator in our lab, that we have the photograph of that you can see here this. So, towards the latter part of this lecture, I will show you some of the results which we have conducted on the treatment of ozonization of wheat grains and its effect on infestation. Okay. So, let us see how ozone works, how it does its disinfestation or its effect on microorganism or microbial killing or inactivation. So, the O3, that is O2 and O. So, O which is attached in O3 to or O2 to form O3, that is in fact still is not very stable. The attachment of O with O2 is quite unstable. So, what happens when the ozone comes in contact with some other biological material, maybe organic or inorganic molecules, etc., then this O part of the O3, which is quite unstable, it combines with the other organic or inorganic molecule with which it comes in contact and causes the oxidation of that molecule, and this oxidation causes the destruction. As you can see here in this figure, that is this ozone, the O part that is one O part of the is attaches itself to the bacterial cell, right, and the O3 reverts to O2. So, O2 is generated formed and O part of the O3 gets itself attached with the microbial cell. It rather dictates the physiological and other processes, and maybe it causes the lysis of the cell components, etc., and the physiological process is affected, and ultimately after some time the cell dies, maybe lysis of the cell takes place. Similarly, when the insects, etc., when they inhale ozone, their respiratory tracts are destroyed, and because of this the insects, etc., get destroyed or they die. Here in this picture you can see the rice grain which is observed with stereoscopic microscope subjected to one atmospheric air in the case of A, and then subjected to ozone gas for different periods of exposure time, and you can see the effect ultimately in these pictures. The ozone has been reported to be effective against almost all varieties of insects which are problematic in food grains, which cause infestation of food grains like wheat, rye, paddy, rice, maize, etc., etc. And this, as earlier also we saw, this ozone, it has a very high oxidizing power. It has a much more oxidation potential than those of the common chemical or oxidants, pro-oxidants or oxidants like peroxides, hyperchlorides, free chlorine, etc. And because of this high oxidation potential, it is effective in the microbial inactivation as well as insects killing. In this slide different researchers have worked and reported that, like for example, Kin et al. In 2003 reported that 120 ppm of ozone in 1.2 days resulted in 94% mortality of Rhizopertha dominica in wheat grain. The same researchers reported that 120 ppm of ozone in 1.2 days resulted in 88% mortality of Tribolium castanum in paddy. So, the various researchers have reported that doses from 50 to around 120 ppm of ozone from maybe 1.2 to 4 days resulted in the mortality of various insects from about 67 to 100% in the grains like wheat, corn, paddy, etc. Means the different insects even can be treated successfully or disinfested successfully. The different grains can be disinfested successfully by optimizing or by using different combinations of ozone and these other factors in the grain, that is the moisture content of the grain and all the things that also. So, and one can get desired mortality. Here it is a system proposed by, it is taken from the literature where some researchers they have given a model, that is how that is in the grain storage bins etc. in the problem, how this ozone can be applied. So, in the lower part of the picture you can see there is a ozone generation unit. So, this ozone it is a conductor that is generated in this system, and then this through pipe it is sent to the storage bin in the bottom of the bin, and from both two sides in this bin the airs are blown. So, this turbulence of air mixes the ozone well with the air, and then this air and ozone mixture passes evenly through the grain, and ultimately that it disinfects the insect etc., kills the insect etc., and then ozone and air safely exits the grain bin. So, this type of setups can be used or can be effectively utilized for disinfestation of grains in the larger storage bins or silos etc. So, this is a case study we are showing in the results in our laboratory. We have done infestation of wheat grain, as I told you earlier, using ozone. We have studied different combinations and permutations, insects also 2-3 common insects which grow on wheat we have used. So, insects of different stages of the insects, all the egg, pupa, larva and adults in different concentrations they were used. Means that is the grains they were first conditioned to varying moisture content, and in this conditioned grains were infested with known number of the different life insects of different life cycle, and then it was stored for some time for allowing the insects to grow, alright. And once means that is in this way samples of under control conditions infested grain samples were created, and of course, the by counting the number of insects etc., by appropriate methods the level of infestation was known, that is what are the number of insects initially present in the grain in the known weight of known quantity of a wheat grain. So, this now wheat grain sample infested wheat grain samples known quantity with the known level of infestation were treated with the ozone gas. You can see here in this bottle, this infested grain samples are kept, and the ozone, it is connected through pipe, the ozone is generated, and this, it is a purely experimental setup. So, it was passed, a known quantity of ozone was passed with this grain for different period of times, and data were collected, and the mortality rate finally was calculated from N0 minus Nf by N0 into 100, that is the percent mortality, where N0 is the initial number of insects present in the infested grain sample, and Nf is the final number of insect after treatment. And these are the response surface graph, that is in this case A, that is it is the effect of ozone concentration and moisture content of the grain on egg mortality, where exposure time and ozone concentration on egg mortality. So, these response says that the moisture content as well as ozone concentration, other exposure time, all these they have significant effect on the mortality of the egg. Similarly, similar effects, that is ozone concentration, moisture content of the grain, exposure time, right, have significant effect on the larva mortality, and even mortality ranging from different percentage, that is even up to 100% of the mortality, ranging from 71 to 100% in this case, where often in other case 57 to 93 or 99%. So, depending upon the conditions of the grain, its temperature, moisture content, concentration of ozone and exposure time, varying degree of that is even up to 100% mortality were found out in our case. This graphs, response surface graph, shows the effect of oxygen, ozone concentration, moisture content of the grain, exposure time on adult mortality, that is adult insect mortality, and similarly the on the pupae mortality. This response surface shows that how the pupae mortality varied with the variation in ozone concentration, moisture content and exposure time and the wheat grain. So, these data, response surface data, were used to develop regression models for the prediction of responses like mortality, adult mortality, pupae mortality, larva mortality and egg mortality, and the R square value or adjusted R square value obtained in this shows that the model is a best fit. In fact, these models were used to predict the values, and then under the optimized conditions the experiment were conducted, and you can see here in the table below that the for all the cases of, that is whether it is adult mortality percentage, pupae mortality percent, larva and egg, the predicted values are close with the experimental value, or experimental values are very close. In fact, residuals are very less, in fact ranging from 0.4 to 2.9 maximum. So, this shows that the models are highly accurate and they are giving their prediction level is very high. Another case study, that is it is taken from the literature, Mason et al. In 2012 they reported that 5 ppm ozone was applied for 5 days to different species of insects in maize. In some time 100% mortality was achieved in 3 days, in some case even 100% mortality was achieved in 5 days, depending upon of course level of infestation, grain conditions and type of the or life stages of the insect. Ozone was applied between 10 to 50 ppm continuously in order to verify it after exposure to ozone. In order to verify it after exposure to ozone, the insects managed to reproduce or not. And in the sample 1, that is 4 days in contact with 10 ppm ozone, it resulted 100% mortality, means the insects were not able to reproduce once after they were treated with the ozone. Similarly, in other case also, that is in second sample, one day in contact with 50 ppm of ozone resulted is 100%. So, it was very effective ozone treatment of grains, whether it is maize grain, whether it is a wheat grain, and it was found to be effective in disinfecting them. So, in our study also from the literature, people have studied and reported the effect of ozone treatment on grain quality. We have analyzed the grain quality, or our results indicated that in fact ozone treatment little bit modified the starch and protein properties, because it might cause the oxidation of starch and proteins, etc. However, the effect are not very significant as far as the quality of the baking characteristics or other characteristics of the wheat is concerned. In fact, some baking properties are positively affected by ozone. Ozone reduces the starch concentration, it results into the lowering of 1,000 grain weight of wheat. However, it enhances the concentration of several minerals and vitamins, etc. Ozone treatment under suitable conditions strengthens the dough, means the protein quality, etc. Maybe this oxidation of protein results has some maturation effect on the protein. So, baking properties, etc. improve. Ozone treatment enhances the shelf life of the grain products. With this, I thank you for your patience hearing. Thank you.